Welcome to my proposal defense. My name is Nin Gan, and I'm working in Dr. Weishu's lab on oysters, and we're studying the effects of the synergistic effects of petroleum pollution and environmental change on oyster health. Okay, so the first thing I want to talk about real quick is about our energy consumption, and I just want to real quick show you guys that 80% of our energy comes from burning fossil fuels, and roughly half of that is due to petroleum. It fluctuates a little bit year to year, but at least in 2019, roughly half of it is due to petroleum. Okay, so because of that and the vast reserves that we have in the Gulf of Mexico, we've drilled like hundreds of these oil rigs in the Gulf of Mexico, coastal regions. And inevitably stuff like this happens, which is the Deepwater Horizon oil spill. And this released millions of barrels of crude oil into the Gulf of Mexico, right? And good? Okay. And then this caused, injuries to countless numbers of birds, fish, and mammals. And this is just the oil slick alone, right? But it turns out the chemicals in the crude oil are toxic themselves. Yes? Good. Question? Okay. So when we study the toxicity of these chemicals, right, scientists usually choose to study these. These are polycyclic aromatic hydrocarbons, right? Polycyclic as in they have multiple of these benzene rings. Aromatic hydrocarbons are ringed carbon compounds, right? So polycyclic aromatic hydrocarbons, I'll be calling them PAH for now on, right? So PAHs, we usually study these when we want to look at the toxicity of these chemicals, okay? Well, it turns out that petroleum is not necessarily the only source, okay? Not just from the raw crude oil itself. These actually also come from other sources such as combustion, right? So if we look at here in the Gulf of Mexico, turns out most of the hydrocarbons from petroleum comes from natural seeps. Well, if we look at this graph right over here, that's just actually the offshore region, right? We're actually more interested in the coastal region where almost all of the human impacts occur, and in that part, pretty much all of it comes from the consumption of petroleum, okay? So most of the petroleum hydrocarbons in the coastal regions come from the partial or incomplete combustion of the petroleum, right? So now that we established this, basically PAHs is not just like oil spill, it's like all of human activity, or 40% of human activity, right, that uses burning of petroleum. Okay, so they're very relevant to our human activities. Let's see what they do, right? How toxic are they? We know that they're very toxic already. There are many studies have done on different organisms, all different types, not just animals. So for example, in bacteria, it causes DNA damage. In birds, it causes reproductive problems. Fish, they may cause cancerous growth. And in oysters, they cause similar reproductive defects and also larval abnormalities. Okay, now I really want to focus on this last group because they're very important to our region right now and our study as well. Right? Why are oysters important? Well, we know basically this is like the talk of this department, right? Oyster reefs. Right? Oyster reefs are super important to our ecosystem. Right? They provide the valuable substrate structure for other organisms to live in. They're essentially ecosystem engineers. They provide food for all the other organisms. Should anything happen to the oysters and the oyster reefs, that affects all the other organisms that depend on it. And also, if the oysters, which are very good bioaccumulators, collect a lot of toxins, then these toxins go up the food chain into other organisms, and then the other organisms biomagnify those toxins and become toxic themselves. And if humans want to try to eat them, then it might be problematic at that point. We should be concerned with the toxicity of PAHs. So that's why in this study, I want to study them, right? It turns out aquaculture is affected as well, right? Since oysters are not just eaten by fish, they're also eaten by humans, and this is a valuable resource, right? We already know that aquaculture, like oysters, are suitable for aquaculture. They are already very used to living in dense congregations. They're very hardy, so they're very good aquaculture species. But not only that, at least in 2017, they provided almost $200 million in revenue, which is the greatest amount for all saltwater culture organisms in the U.S. So in terms of economic revenue, any problems facing oysters, it could even have some economic effects at this point. And this is even more relevant now that aquaculture is expanding, and now even in Texas, we can start doing aquaculture, right? The second longest coastline in the Gulf of Mexico, that probably provides a lot of new space to do aquaculture. So are the waters around this area suitable for aquaculture? Well, are they going to, be good for the oysters? Cause like healthy oysters that are not contaminated? Well, we'll see, right? So that's why we're trying to study the effects of the PAHs on the oyster. Now this is a topic that has actually been very well studied already, because PAHs are popular and so are oysters, right? PAHs, like we already know, that's basically 40% of human activity. And oysters are popular for studying toxins in the water because they're so good at bioaccumulating. So why are we focused on this? Well, it turns out that, PAHs, they don't just stay the same in the environment. There's a lot of things going on in the environment that could change the PAHs in some way. So we want to study those changes basically, right? So for example, we already know that, oysters are affected by PAHs in these examples. Let me just read them off real quick. This, in addition to the one I've already went over, they cause some pre-larval defects. So in the gametes and the embryos, they also cause some like immune problems, so that weakens the immune system, and then this causes it harder to stave off disease or parasites. And then they also cause upregulation of several genes associated with some oxidative stress response, either detoxifying enzymes. And then they also cause actual tissue damage and DNA damage as well, right? So we know this about oysters. Let's move on, right? What happens to PAHs in the environment, like I mentioned? Well, it turns out there's sunlight, right? Sunlight has UV rays, provides activation energy enough for it to possibly degrade into other compounds, right? Why is this important? Because a few years ago, Dr. Xu ran this experiment of a different organic pollutant, dichlorine. So this is not a PAH, but it's still organic pollutant affected by UV light. Turns out that this chemical causes, the reduced heart rate and upregulation of stress proteins such as defensin, calmodulin, heat shock protein 70, just from dichlorine itself. But there's another treatment, a treatment in which this chemical was treated with UV light, and it caused even more of that damage. So now maybe we need to start paying attention to the photodegraded versions of these organic pollutants. Okay, so a quick look at what were some of the chemicals found in the photodegraded mixture. We found quinones, right? Benzoquinone to be exact, and we know that quinones are highly oxidative and they're very toxic to organisms. Okay, all right. So what happens to PAHs? PAHs under sunlight, very similar, right? We have found that they turn into their own PAH quinones, oxy PAHs, nitro PAHs, hydroxy PAHs, and some aldehydes as well. You guys look at the chemical structures of some of these. The extra hydroxyl groups, the carbonyl groups, make these compounds way more oxidative than their parent PAH molecules. Okay, so this is what primarily we're going to be looking at. Should they arise when we degrade them with sunlight? Okay. This is the case in other animals, right? This is what we have found. Like there's very little literature of these compounds on oysters so far, but we can see they're very toxic, right? Like zebrafish is a very popular model for toxicology, and it turns out that oxy PAHs has caused a lot more DNA damage than the parent PAHs they came from, right? Down here, copepods, little crustaceans, they're affected as well. Experiment done, the nitro PAHs, they cause 1,000 times more reactive oxygen species formation, right? That's the bad kind. Those are free radicals, right? They are highly oxidative to tissues. Well, 1,000 times more of those were produced in the light experiments versus the dark experiments. There's got to be some sort of thing that's happening in the light that we need to be wary of. And it's not like light doesn't exist, it's very ubiquitous, right? Okay, so we're interested in not only studying the PAHs, therefore, we're actually more interested in studying the environmentally altered PAHs, more specifically the photodegraded ones on the oyster health. So how are we going to do that? Well, we're going to follow the previous example with the dichlorine. We're going to make the oyster cell culture right? It's really cool. We're going to dissect the oyster. We're going to locate its heart. It's pretty easy to find. It's in the chamber next to the adductor muscle, right? We're going to culture the heart cells. Why? Because they're very convenient, right? The oyster heart, it has a very telling and obvious physiological sign, right? It's heartbeat that we can use to measure possible toxicity levels of the chemicals it's exposed to, or we expose them to. Okay, so we're going to be measuring its heart rate, and then at the end of that, we're also going to be looking at its gene expression using qPCR, and I've chosen these genes, defensin, calmodulin, and heat shock protein 70. They're, pretty general stress proteins, so like defensin used for like immune response, calmodulin, which is used for, it's a calcium binding protein, so it's pretty useful in all like bodily functions. Heat shock protein 70 is a pretty generic stress protein related with environmental change. So we're going to be looking to see if any of the expression of these is changed or increased, especially if we apply these photodegraded PAHs to these oyster cells. We can take this study a step further by not only looking at the heart, we can look at the mantle as well, right? The mantle is responsible for making the shell, so we can actually study the process of biomineralization and how it is affected by these compounds, right? For that, I've chosen these proteins, right? The gene expression of amorphous calcium carbonate binding protein, which is used for depositing calcium into its shell. PIF80, which helps in calcifying the nacreous layer, and also nacrein, which is carbonic anhydrase, which helps. There's a bunch of them, but this one especially helps regulate the carbonate concentration inside its hemoglobin. Okay, so we can do really similar studies with the heart and mantle, but we can look at these two processes, see how they are affected by PAHs and the photodegraded compounds. Okay, so when I say photodegraded, I just want to remind you guys, it means we're exposing them to sunlight. Okay, so this is the specifics of what I'm going to do. We have chosen 3 PAHs because of their relatively well-known toxicity levels. Okay, so we have anthracene, pyrene, and dibenzothiophenone, right? And we're also going to have a mixture of those. And then we're going to have concentrations of about 1 ppm or the maximum, because they're not very soluble, so we'll see what we can get, what concentrations we can achieve. And we are going to be exposing them to artificial sunlight in a solar simulator right here called the sun test for anywhere between 0 and 24 hours within that interval. And then we're going to take that solution that has been irradiated and expose, the cell cultures for 24 hours. Okay, and then after that we're collecting our data, like I mentioned, the gene expression, the heart rates, and then we're going to analyze all the data in R. Okay, we're going to be doing 1 or 2-way ANOVAs depending on how many factors we're trying to compare. So if we're looking at just the compounds between the different ones, which one is the most toxic, then 1-way. And if we're trying to find interactive effects between the type of compound and how much exposure, sunlight exposure time, then we'll be doing 2-way ANOVAs for that. So overall, my experiment number 1 looks like this. Okay, we're going to have our cell cultures, right? The cell cultures is what we're going to do our experiment on. We're going to be treating them with PAHs on the left, right? Those PAHs are going to be irradiated with the solar simulator. They're going to be treated, and after that, we are going to be measuring its heart rate and also gene expression. We're going to be doing qPCR on those select genes I mentioned before. Okay? All right, so what do I hope to get out of this? Well, I want to see what's inside those photodegraded mixtures first of all, and this is probably something that Dr. Armbrust is going to help us with. Next, we are going to quantify the toxicity of those PAHs and the photodegraded versions, right, the products from that. We're going to see what are the relative toxicities of those, okay, compared to one another, and also the photodegraded versus the parent PAHs, and see if they actually are more toxic as we expect them to be. Lastly, I want to build some linear models that relate the photodegradation time to the toxicity. Okay, so what have we done so far? We got these cell cultures. They're pretty cool. This is day 1 after seeding the heart cells, and they look like that. all the viable cells have attached to the bottom of the cell culture plate at the time. By day 2, they migrate into these cell cultures, and by day 6 or the end of the week, pretty much, they start eating, right? Okay, so this might be the ideal time to begin the treatments. However, I noticed some interesting things, right? Counting the heartbeats of the untreated cells thus far yielded very high range, between 10 and 60 beats per minute. And that's really variable. Maybe I'm gonna have to have a really big sample size. But I think maybe there's a lot of other factors that govern the heart rate. So like maybe the shape or the size of the cell cluster. So I'm thinking of collecting additional parameters to try to reduce the variation in that. I've also found a very slight but still significant upwards trend in the first week after they started beating. And this could be problematic if there's an additional variable to consider, time, right? So if this is real, I don't know, maybe it looks like it's starting to level off. So I think that if I just collect some more data on this, and if it does level off, then I can find a suitable day to begin treatments. If not, I'm gonna pick a day and standardize that with all the other tests. Okay, we're just gonna use the same day each time. Okay, so that's our first experiment, and it is expected to be completed by the end of the summer. Okay, now moving on. There are not just sunlight in the atmosphere, in the environment, right? There's a lot of other things. We project that, rising PCO2 levels in the atmosphere, they probably, create more dissolution of carbon dioxide in the ocean, affects the carbonate chemistry, makes it harder for calcifying organisms to biomineralize, right? Well, if that's the case, it makes it harder. Well, oysters biomineralize, right? All of a sudden, this biomineralization process, which is a normal process, starts to, become like a burden, right? It starts to compete with the resources needed to help it defend itself from toxins and stuff, right? So this could affect the oyster's response to the pHs and the photodegraded products, right? We want to see how changing pH levels affect this, right? So a more acidic environment could do one of two things, right? It could change how the pH degrades, first of all, and second of all, it could affect the oyster's physiology to see to change its response to the toxic chemicals. Okay, now that's one thing, right? That's like a future projection of changing environments. Well, here's like a present day, like naturally fluctuating environment thing. We know that oysters live in the bay that is subject to a very wide range of salinities, and this changes, right? Because periodic runoff and also proximity to the ocean, the salinities that the oyster is naturally exposed to is very different. So we're going to check that one as well. Differences in salinity could change how the pHs photodegrade, or they could change the oyster's response to the photodegraded pHs. Okay, so we're going to be looking at what effects those could have on the oysters as well. Okay, so what are the specifics? Well, the pH, we have 3 different pHs that we're going to check, right? The normal one, pH 8.2, and then we have 2 kind of extreme ocean acidification scenarios, right? So we projected PCO2 levels in the atmosphere and predicted pHs that occur from those. Predicted pHs that occur from those projected PCO2 levels, right? Salinity is going to be between 10 and 30 parts per thousand, which are normal salinities we could see in Corpus Christi Bay around this bay. Okay, the same thing, analysis. This time we're going to be doing 2 ANOVAs because there's an extra factor now. Okay, we're not going to check pH versus salinity. We're going to actually conduct those tests separately, right? As I'll explain in my experimental design. We're just going to be looking at interactive effects between pH and the exposure time, and then salinity and the exposure time on the toxicity. Okay, so this is experimental design I was talking about. We have 3 groups now. A base group, in which I don't alter the environmental conditions, right? A pH group, we're going to be altering the environmental conditions, just pH, and this one, just salinity, okay? So we're not going to do any in which both the salinity and the pH is altered. There's too many groups if we do that. We're just going to keep it like this for now. Maybe that's a future one, who knows, right? Okay, as before, we have our pHs. We have our photodegraded pHs. We're going to apply them to these 3 groups, okay? And and we're going to measure the heart rate and qPCR for gene expression. Okay? All right, so that's basically our experiment number 2. As if you guys didn't just notice, the base group is actually experiment 1, so experiment 2 is these 2 parts. Right? Okay, and so what do I hope to get out of this? Well, we want to see how pH and salinity affect the toxicity of those chemicals, right? We want to build linear models to quantify those effects, and we want to find evidence of interactive effects between the environmental conditions and the petroleum compounds, right? So this is basically the synergy part, right? Well, we want to see the synergistic effects of environmental change and also petroleum pollution on the oysters. Okay, so that means that our objective is now not only to look at pHs and the photodegraded pHs on the oyster, but we want to see what happens to the oysters under these conditions in different regions, right? So that's our different salinities, and in future ocean acidification scenarios, right? So we're going to be basically doing this, but under those 2 different conditions. Okay, so that's my experiment 2, and I expect it to be completed by the fall. Okay, so far I've only talked about cell cultures, right? But we know that an oyster is not just a cell culture, right? It's not just one organ. Many organs working together as a shell has many defensive capabilities against contaminants. So we're going to be looking at actual oyster individuals as well. Okay, this is the final stage, looking at the oyster individuals. And the perks is they got different life stages, right? So we can look at the differences that are affected by the life stage of the oyster. We're going to be obtaining some larvae, possibly 48 hours post-spawn. Post-fertilization causes them to become D-shaped larvae, and they're very easy to obtain. If we wait any longer, there might be too high mortality, but if we use them right at this time, we'll be able to actually generate a lot of them. Juveniles and adults, right? Juveniles 1 centimeter, adults 12 market size. So we're going to see how their respective age group cohort affects them. Okay, so we're going to do the same thing as before, right? We're going to have our 3 groups, the environmentally altered ones, pH, salinity, and our base group. We're going to have the same chemicals, the same solar simulator, and we're going to apply them. However, this time is different because there's actual oysters, so there's a lot of data we can get from each oyster, right? We can get bioaccumulation data, stress response, actual tissue damage, and mortality, right? So let's go over each one of these. Bioaccumulation data, right? We want to see what kind of chemical actually ended up in the oysters. So we dissect the oysters and try to get the organs that usually bioaccumulate the most toxins. Okay, so that's going to be the mantle, the digestive gland, and the gonads. Right? The high lipid content usually has a lot of toxins inside of those. Okay, next, we're going to be looking at the stress response, but this time there's going to be a lot of different things we can look at, right? Various tissues throughout the body for soul, not just the heart. But look, we can look at all these different proteins, right? Defense and calmodulin heat shock protein 70, we've already saw those as generic stress proteins. But we can also look at stuff like pyruvate kinase, citrate synthase, and the cytochrome C oxidase, right? Those, if you just like hear those names, like remember pyruvate, citrate, those are like the 3 basic steps of cell respiration right there. So those are metabolic enzymes. So we can look at how the metabolism of the oyster is affected, right? We expect something like increased metabolism in the initial stages, probably to try to maintain homeostasis, and then it may decrease later on if the toxins start inhibiting those processes. We'll see. Okay, and then the last one, we have a catalase, glutathione peroxidase and glutathione reductase, and also superoxide dismutase. Now those are detoxifying enzymes. It's really important because if these chemicals really are toxic and oxidative, then we're going to need these, right? Superoxide dismutase is basically the enzyme that's responsible for breaking down reactive oxygen species, and once that happens, it breaks it down to hydrogen peroxide, which is still oxidative. So we need catalase to do that too, right? And then glutathione, reductase, and peroxidase, those are for breaking down quinones. So look, if these are indeed toxic, then these are all going to be used by the oyster to try to protect itself. And then we're going to see if it's toxic, there's going to be some sort of detoxification response. Okay, so we're going to look at those enzymes as well. Lastly, thirdly, we're going to be looking at actual tissue damage, and for that, we're just going to look at the tissues, right? Put them on the microscope slide, histo slide. I've identified a couple different organs that are suitable for this. Right? Past studies have shown that the digestive gland, the tubules, may atrophy in the presence of toxic chemicals, and also the gonads. Right? Reproduction is a process that requires a lot of energy, and oftentimes it is delayed if the energy needs to be diverted somewhere else. So oocyte atresia, which is basically the female oyster trying to release oocytes, but then it reabsorbs them, just skips it. This is what is shown in this picture. It's kind of hard to see, but oocyte reabsorption, we could look at those things to see actual tissue damage, physiological effects going on in the oysters exposed to these chemicals. Okay, and then lastly, of course, we can look at mortality. That one's pretty obvious, right? We're going to check the mortality within a period of 4 days. Okay, so once we get our data, we're going to analyze them. We're going to do principal component analysis to determine what are the most important factors. Right now that there's a lot of factors, we determine what are the most important ones, and once we have identified those, then we can do two-way analysis to see which of those components and how they interact to affect both bioaccumulation and toxicity. Toxicity is measured in either a mortality or the gene expression. Okay, and then, so this is my experiment design for a third one. It's pretty similar to the rest, right? The only main difference is we're using actual oysters. There's going to be different life stages, and they may cause some variation, and then we are going to check, the bioaccumulation levels, the qPCR for the gene expression, actual tissue damage, and of course oyster mortality, right? Okay, excuse me. So what do we hope to get out of this? Well, we want to see the sensitivity of each of those life stages, right? How does the life stage affect its response to photodegraded PAHs? How do those life stages respond to the pH and salinity changes, right? Maybe one of those will, maybe even if the photodegraded PAHs cause the same damage in all three of them, it could be the case that these things may cause, variable damage depending on life stage, right? So we're going to be looking at that one as well. We're going to build a linear model relating the bioaccumulation to toxicity. So just like tissue concentration of the chemicals and seeing if they're correlated, right? And lastly, we're going to see which of these factors, right, and how they are related to either bioaccumulation or toxicity. Okay, so hopefully that makes sense so far. The last one is, remember we collected mortality data, so we can create a survivorship curve over time, especially for the larva. This is probably mainly just for the larva. Okay, and so our final tally for objectives is not only just to study the photodegraded pHs, not only just to study them under the changing environment, right, different regions, but it's also to see how the different life stages factor in and how they're affected by these two processes. Okay, so hopefully that all comes together for you guys, and this final stage of the experiment is expected to commence and finish in 2022. Okay, so anyways, at the end of this, I want to just quickly go over my broader impacts, and I just want to remind you guys, oysters are really important in the environment and for aquaculture, right? In the environment, they are ecosystem engineers, and for aquaculture, they're very popular. They're easy to grow compared to other animals, and they bring in economic revenue, right? So should anything happen to them, then that would be a bad thing, right? That would add incentive for either the reduction of pollution, or better yet, like we just attack the source, right? We know that the source of the pHs actually mostly comes from the usage, so use less, right? Less of the combustion. So hopefully we can try to add some more incentive towards that angle, right? Second thing, in regards to aquaculture, we don't want to grow contaminated oysters. We don't want to fail at growing because the area kills off the oysters. So we can use the bioaccumulation abilities of the natural oysters that exist to see if certain regions are suitable for aquaculture, right? Now that we know how the toxins affect the oyster, we can see if the oysters in the area, are suffering from a very high load or a reasonable load. And lastly, suppose that the pollutants are persistent and the environmental change is inevitable, then perhaps this information could be helped to be used to develop new strains of oysters that may be tolerant to these conditions, and we'll still be able to do our thing regardless. Okay? So that's the end of my presentation of my proposal. Hope that made sense. Hope I didn't talk too quickly, but I would like to thank Texas A&M University Corpus Christi for the program and the facilities, and SF for the funding, my lab members, especially Dr. Shu for the ideas of the project and for all the help he's given me, and of course Sandra, Molly, Leisha, and Zane Landrew for their help in taking care of the oysters that we hold in the lab. Okay, so I guess now I'm going to take some questions.
Thanks, Nin. Actually, before we start our question, I want to introduce some of the committee members that Nin has. Of course, I'm Newt's advisor. I'll be serving as the chair of the committee. And in the committee, we also have, in the room, we have Dr. Xiping Hu, who is a faculty member in the physical and environmental science, and also a HRI chair in ecosystem science and modeling. Okay, and he's interested in studying the ocean acidification in the ecosystem. And we also have Dr. Chris Holloman sitting here. Is one of the youngest faculty members here in our department of life sciences, and he's interested in studying the oyster genetics. Okay. And we also have another committee members at Louisiana State University, Dr. Kevin Ambras. I believe some of us might already seen your picture from the website, and he's the chair of the Department of Environmental Sciences at the LSU, and he's been studying the toxicities of a wide range of the pollutants, including the agrochemicals, the petroleum-related pollutants, a very wide range of the materials. So this is Nin study was actually supported by this NSF project, which is a collaborative study between our lab and Dr. Ambrose lab. So, and with that, I think we have a couple of minutes maybe open to the general audience. So whoever has questions at this moment, you can feel free to ask. And also, if you wanted to type in your question in the chatting box, we can also look at it. So you guys only have 5 minutes. Any questions? Last chance. I guess you made everything clear enough.
Thank you.
Okay. If that's the case, we're going to start the question from the community members. So, who want to be the first? Should we want to start?
Thanks, everyone.
Sure. No, Kevin.
Dr. Ambros, you're me.
Kevin, are you there?
Yeah, why don't you let somebody else go first? I'm having a few technical issues over here. Let me see if I can sort these out.
Okay.
No problem.
All right, sure. Shipping, it's you. Or Chris.
I have a question.
Yeah.
So, the levels of PAHs that you're testing in your tissue culture, I think you said 1 part per million.
Yeah.
How does that relate to an environmental level of some sort? Is that background level? Is that this is just something I don't know anything about? Is this oil spill level?
I'm going to repeat the questions. Yep, got you. Okay, so Dr. Hollenbeck asked a question about why 1 part per million was chosen as a dosage for the heart cells. Okay, so here's the thing: inside of the oyster, right? So that's actually a tissue concentration, okay? So there's other studies that show that tissue concentrations may reach those levels, but also we're just trying to get as much as we can because they're very insoluble. So we kind of chose that. But you know, like for the most part, it's like a tissue concentration because in studies that have done bioaccumulation studies on oysters collected from contaminant environments, they may show levels around that level. A little higher because they're actually looking at the solid tissues, and that one's kind of like a hemolymph concentration.
So a living oyster could have that tissue
Could have that,
concentration.
yes.
But how do you control it just show 1 ppm in the tissue? What would be the water, the pH?
Oh, okay. No. So we're gonna make the cell culture medium. Oh, sorry. The question is like, how do we control to make sure the tissue has a concentration of 1 ppm? We're not. We're going to just make the cell culture medium 1 ppm, and it's going to accumulate on its own. Whatever concentration that is, that's left to be seen.
And then if you compare, you have 1 ppm, then you have the maximum level that is unknown. And how can you tell whether there may be a concentration in the middle that is toxicity relevant?
Oh, okay. So the question is about why there aren't more toxicities. I personally don't think that we should focus on dose for this study. I think actually more importantly, we're focusing on the photodegradation times. So if we simplify our study a little bit with only 1 dose, 1 or 2 doses, and different doses, and mainly focus on the effects of the photodegradation time. Like we can look at dosage experiments too, but I think more important we should focus on that. If we have time, I guess. We can include something like 0.1 ppm as well as 1 ppm.
Do you have any reason to think that dosage might be on the far end of toxicity, either not toxic to anything or too toxic, where you wouldn't have any variation?
Oh, I see. I guess we're just gonna have to figure that one out. Oh yeah. So the question was basically asking whether 1 ppm might be too high and we won't see any variation. That's very interesting. I think we're gonna have trouble dissolving the PAHs to begin with. So we're just kind of like aiming for as high as we can. If they do, obviously, if they do end up killing everything, then we're gonna have to reduce the dosage, and then we'll just change it to 0.1 or less.
Also, for your treatment, you have multiple pH compounds.
Yeah.
And are you going to test all them as a mix or just individually?
Okay, so the question was about the different pH compounds and how I'm going to administer the treatments. So they are going to be individual and there's going to be mixture, so there's 4 groups.
If it's a mix, then what will be, because different, from the chemistry point of view, different compound may have different toxicity. If you have a mix, what will what's the proportion for each?
I think 2 possible ways we can go about this is either equal proportions or look at natural levels. Yeah, so we'll probably decide between one of those 2. But I mean, we kind of want to see the difference in the toxicity for the different chemicals, and so equal proportions might be a good way to do that.
I think you got something from Kevin.
Okay.
And you also mentioned the solubility will be the issue with this.
Yeah, okay, so the solubility will be an issue with trying to get the equal concentrations, I'm guessing?
Yeah, I think this is a point that we've already discussed. For most of the chemicals, most of the pHs, although he put the number 1 ppm there, it's almost a lot of the pHs cannot even reach 1 ppm. So that's why he also had the kind of another side note saying, or the maximum solubility. So that's something we can't really change. But just the fact that we're really looking to is really the accumulation. So that's why if you can open up a oyster collected from the field, most likely you're gonna find the concentration of the PAHs inside the oyster is way higher than any area in the ocean. So
Yeah.
that's something like we consider, but we have to figure out what's the best the concentration series we want to use for that.
Okay, thanks for the explanation. Thank you, William.
Yeah, let's go through the questions I had. Why use hard cell?
Because they have a convenient physiological.
You have to repeat the question.
Oh, okay. Why are we using the heart cells? Because they have very convenient physiological effects such as the heart rate. That's one thing. It's easy to quantify. The second thing is, like the blood goes through there. The blood has a lot of the proteins that we're trying to look for. So there's a lot of hemocytes and the hemolymph that goes through the heart, and it also goes through the muscle tissue. But like if we're trying to quantify all those different types of gene expression, then we want to look at where the genes pop up the most.
And then for this hard cell, I'm not totally not biologist at all, but if you put the cells in the medium, how without any kind of stress, how long can they survive?
Okay, so
How do you maintain the cell to be healthy?
yeah, the question was about how long is the heart cell culture viable for, and so the previous studies can do this for maybe like 40 days or so, but so far in the ones that I have made, they're viable for at least 2 weeks, long enough to do the experiment at least. But I have one going on right now that's 3 weeks old and they're still beating, so that's good. How do we maintain their health? By changing their medium once every 2 days, maybe more often. But I kind of like one every 2 days because that way there's a day in the middle in which I can collect data.
If you have the hard cell, if there's a stress, would it be faster or slower? Which one would be the criteria for being stressed?
Short-term acute is probably going to be faster, and then wait a little bit is probably going to be slower after the damage sets in. Yeah, that's just expected. But who knows? We'll figure that out. Yeah. Oh, sorry. The question was whether the heart rate will be greater or lower depending on the stress. So, in general, stress causes the heart rate to go up in acute situations, but then after longer term, it starts to go down because the chemicals could either cause the oyster to increase its metabolism eventually to try to cope, but then eventually the chemicals start to have inhibitory processes which eventually slow it down. So maybe probably increase and then decrease. Is that?
Yeah, sure. But is it closed already? I think still somebody online. There's still online.
Well, maybe they wanted to listen to the questions. So we have a couple more people here. So I didn't really mention, but at this moment, all the questions are supposed to be like closed door questions, just specifically from the committee members. So thank you all for coming
Thank you
for the general audience. So now you can do whatever you're planning to do next. All right. So, but I can't make them out. Okay. Any other questions?
Well, you're a Louisiana community member.
So Kevin,
Dr. Russ, do you have any questions by now?
Yeah, I've actually got a few.
Okay.
I've got a couple here. Actually, more related to the actual design of what you're trying to do. Okay, for experiment 1 in your cell exposure with the sun test unit, you didn't mention, and this is just sort of a logistical thing from my experience working with these, those things generate a ton of heat, and you could end up,
Evaporating.
putting cell cultures in there, you're effectively gonna end up having
Oh, I see.
mush cultures, unless you're controlling the temperature. How are you planning on controlling the temperature in that?
Oh, okay. I'm actually not going to photodegrade the compounds during the treatment. So we're not putting the cell cultures in the sun test. Yeah, sorry. I guess I should have mentioned that.
The picture kind of looked, from the picture it kind of looked like it was, and I'm a little concerned about that.
Oh, okay. Well, I mean,
Cooking it.
the sun test was in a separate box from the cell cultures. Yeah, so the sun test is applied to only the PAHs, and then they are removed, and that solution is saved, diluted, and it's going to be treated. We're going to treat the oyster cell cultures by adding it to the medium.
Okay, yeah. Yeah, so I was gonna say, unless you're planning on controlling temperature, you're gonna end up with a mess trying to do that.
Yeah, definitely. The cell cultures need to be at 28 degrees.
Okay, for the pH, you said you're gonna be bubbling CO2, you're gonna be changing the pH by bubbling CO2 into this. I mean, obviously, we can see ocean acidification is part of the responsibility for ocean acidification is through increased CO2 in the atmosphere, and then going through the typical carbonate cycling, as you showed, but at least experimentally, if you're just really looking at the effective pH, why change it by bubbling CO2? Why not just manually adjust it with hydrochloric acid?
Oh, I see what you're saying. Okay, so I guess.
Life a little easier.
Yeah. Okay. I guess I should have rephrased my presentation. So I apologize for this. I thought that pH would be more easy to understand, but we're actually not really testing the effects of pH. We're actually testing the effects of increased PCO2 levels. So we're gonna have 3 different PCO2 settings, and then the pHs that I provided were predicted pHs based off those projected levels.
Okay.
So we're actually we're really interested in the natural process of the CO2, so not the acidity itself. Okay, so we have 3 different levels of carbon dioxide we're trying to add: 400, 1600, and 2800 TPT.
I'm assuming.
Right? I think it's PPT.
Yeah.
ppm. Yeah, ppm. Right. ppm in the atmosphere, and that will result in approximate pHs that I mentioned in here. So yeah, we're actually testing PCO2. Yeah. So that's the clarification. Sorry for the confusion.
Okay, so the end point, the thing you're testing is the PCO2 level, not the result, not the actual pH itself.
Yeah, PCO2, not the actual pH.
Okay, then that makes sense. And I think we already talked about the part per million per site causing the issue of solubility.
Yeah.
The other thing I'm thinking about, and I'm kind of just thinking, I'm thinking aloud here. So, and I haven't made a formal decision about this, but we're talking about testing the PAHs directly, and that's obviously going to cause issues. One of the big challenges is going to be the solubility piece on it, as we discussed already. But one of the general hypotheses is that some of the impacts of these are, that they're running through a very similar, people have looked at photodegradation of these things before, and they see that you see increased hydroxylation of these, and of course, especially if you start having indirect photodegradation processes, those are, if they're radically driven, that's typically an electrophilic process. And the more hydroxylated these become, the more susceptible they are to electrophilic attack.
Okay.
But that being, and then of course the more hydroxylated they are too, the more susceptible they are to direct photodegradation. So if that's the case, I'm wondering ultimately, one thing I didn't see in your proposal when I was reading through this, and when I was reading through it fairly quickly, one thing that, and this is against one of the ways we do this here at LSU, but one of the things I typically like to see in these perspectives, I saw that you had your goal, your objectives. I didn't see I usually like to see a series of typical hypotheses that you're trying to test too. You know, overriding hypotheses. And that's something that would possibly be good to add to this.
Okay, thanks for the
And we
feedback.
can get, you know, the rest of the committee can chime in and see what they think on that,
Okay.
but that would help guide some of this too, I think.
Okay, thanks for that. I do believe I have the hypotheses, but they may not be as clear as they should.
Yeah, like I said, I did this when I was reading through this fairly quickly yesterday, so.
Yep, no problem. But thanks for that suggestion.
Yeah, but it may be, I brought you, again, you know, like I said, I'm thinking of it off the top of my head as I was going through this, maybe, it may, you know, if we start with this solubility problem starts becoming a bigger problem, we may want to step back and do that.
I see, okay,
That
so yeah,
is-
if we can't
the
generate.
environment, that's what's actually driving some of this, that could be the case. There's a lot of questions too, even with some of these, there just hasn't been a whole lot of work done with hydroxylated PAHs. And in theory, those are the things that would be driving some of the greater parts of exposure, if that actually does come into play, so.
Yeah.
I don't know, like I said, just a thought.
Okay, thank you for that. Yeah, that definitely could help because, I mean, the reason why a lot of those compounds are more toxic is not just because they're oxidative, but also because they're more soluble and bioavailable. So, you know, that could be something we could do. However, I think at this time we're still gonna try the original. We want to see how the pHs photodegrade by themselves because, you know, like if we get into a hydroxylated pH study, then basically that just becomes a dosage study. And right now we're trying to figure out the difference between sunlight exposure times. But yeah, thank you for that suggestion.
Any other questions, Kevin?
No, that's it.
Okay. Well, thank you, Dr. Armbrust. It's good to hear.
You did a good job pulling that together.
Oh, okay.
It was a nice presentation.
Thank you. Yeah, this is my first time talking to you, so it's good to hear from you.
Well, hopefully we'll be able to do this thing in person, so I'll be able to come down here and we'll do this in person sometime.
Yeah, yeah.
Hopefully soon. Should be very soon. Any other questions?
I have a couple of questions.
Okay, Dr.
Just
Holmes.
sort of practical things. How are you planning to do the to measure larval mortality in a time series sort of way?
Okay, so it depends on how small they are, right? So, one way we can do this is, you know, we have like larval counting devices like hemocytometers, or, you know, we just put them, or if we don't use that, we can even just put them on a normal slide and count the number within you know, the density within the frame. You know, and that'll be it because the density, you know, like that'll be the number we'll be comparing. So, how many, like all the dead larvae should have sunk to the bottom, I'm guessing. So, you know, all the samples of the water samples we'll be collecting will have the live larvae in it. We put it on the slide and we can just count them on the microscope.
Yeah, that was my question. Do you think you could tell a live larvae from a very recently?
I think so
within
because
the course of several hours.
yeah, I actually do think so because the dead ones they like break. You know, they're like broken. Yeah, so I think it should be. And then, like I said, if the dead ones, you know, sink to the bottom, then if I collect it from the middle of the water column, it should just be the live ones swimming.
So what I found, this is a little bit difficult because you have to, when you're counting, you have to mix the water well in order to get a representative sample. And when you mix the water well, you you stir everything up, and so you have to be able to visually tell, you know, what's an empty shell from a a live larva, and it's easier if you're looking over the course of several days because the live ones grow faster.
Mhm.
So
Yeah, they're bigger.
so they'll be
Oh,
smaller
okay.
dead shells. But
Yeah,
if you're
that's a
looking
good suggestion.
over the course of a day or 2 days, I think it might be tricky. You might have to refine your technique a little bit.
Oh, I see.
Is there some kind of live dye that we could use to dye it?
Probably so. Yeah, I bet
Yeah,
that.
I think that would be easier, right?
Uh-huh.
Oh, okay, cool. So like,
So if you do it.
has like something in the body that
Like
reacts with
blue,
it.
yeah, something
Okay,
like that.
but then the dead ones don't have that.
Yep, that'd be a good way to do it.
Yeah, okay, cool. Thanks for that suggestion.
And then
Yeah, I didn't realize it was how problematic it might be.
Yeah, they're just so small. It's hard to
Yeah.
hard to work with. My other question was, have you considered the genetic background of any of the oysters that you're testing?
Only a little bit, so.
Repeat the question so then we can.
Oh, okay, so Dr. Hollenbeck asked a question about whether we consider the genetic background. And, okay, so ideally, we want them all to have a very similar genetic background. Okay, so that there's no like, oysters that are just genetically better than other ones at withstanding fluorinated pHs. So we are planning on trying to get our oysters from the hatchery. So, if they use the broodstock and then they create the larvae, then, they create the juvenile oysters, then they should be all from the same cohort. So, more
So-
or less related.
I think that's a good way to do it. But for your cell cultures, even if you're getting live oysters from, so the really interesting thing about where we're at is if you get live oysters from Corpus Christi Bay or even Aransas Bay,
Yeah.
they're two very distinct subspecies.
Okay.
They're all
That's good to know,
they're all C.
actually. I didn't
virginica,
know that.
but in sort of equal proportions. In Corpus Christi Bay, it's about 50% of each, and they're very distinct. You can't really, because they sort of
You mean they're not very distinct, so it's hard to tell.
they're not distinct morphologically, but genetically
Oh, genetically they're
they're
distinct.
completely different. So the South Texas group is very different, and the group that we have in North Texas is more similar to what's on the East Coast and up into Canada than what's in South Texas. And so you could, if you sampled from here or from Rockport, or if you source local oysters, you have probably an equal chance of getting one of the two, and they're so different that I would say you should definitely test them. It's an easy.
Oh, do a genetic test?
Yeah.
Oh,
Yeah, I think that's probably also explain why you have a great variation for the cell culture at this moment.
No, I don't think so.
Well, if you have a similar genetic background, you might get something more consistent.
It could be, but I
Yeah.
was thinking at least for gene expression, you might get two different groups just because of background
Yeah,
genetic differences
okay. Yeah,
than something experimental. So,
I see.
but the test is inexpensive, so
Okay.
I can let you know how you would want to do that if you wanted to
that's
test
what you're
everything
here.
after the fact and just say this was of this type and this was that type.
Yeah.
That would help, I think.
Cool. Can you identify them by your eyes?
No, it's
Just
just
genetic.
they're so. I brought a figure of something we just generated that shows you how different they are
Can you show me?
genetically. So this is.
Oh wow, they're like orthogonal.
Yeah. So this is a PCA.
Yeah, they're completely different.
Of about 10,000 sample size.
Yeah.
This is all South Texas. This is North Texas, and these are some other places in the Gulf. But if you took 50 oysters from the University Beach, half of them would be here and half of them would be here. So they're completely.